In this video, we'll talk about eukaryotic transcription. We would look at the nitty-gritty details in this video, so stay tuned till the end. Transcription is the process of copying a segment of DNA and making a RNA out of it. So the RNA is the blueprint for making protein or sometimes also making RNA, and this process is known as transcription. Eukaryotic transcription is much more complicated than prokaryote. This particular transcript is produced many in number. So DNA is read several times by the reader, which is the RNA polymerase, and several copies of mRNA is generated. In this video, we would talk about the steps of eukaryotic transcription, that means initiation, elongation, and termination. We would look at the factors that regulate eukaryotic transcription, such as transcription factors, activators, repressors, and many others. Then we would talk about the chromatin-centric view of eukaryotic transcription. And lastly, we would look at the techniques to study transcription in vivo. So let's start with the conventional way of looking at this transcription process. Everything starts at an initiation step, and everything starts at the core promoter. So RNA polymerase 2 has core promoter element, and the most characterized core promoter element is the TA box, which has several thymine and adenine nucleotides in sequence. So the TATA box is bound by a transcription factor, which is a general transcription factor of TF2 family. This is known as TF2D. This has two domains. One is TBP, the TATA box binding protein. It binds to the DNA and distorts the DNA eventually, and there is a TBP associated factor which interacts with other components. One of the peculiar features about TF2D or TATA box binding is basically it creates a distortion in the DNA and bends the DNA almost around 90 degrees. So after TF2D, there are other factors which would be recruited. But TF2D, unlike other transcription factor, don't have alpha helical structure in the DNA binding domain. Instead, it has a beta pleated sheet, and all these kind of structural information was derived by X-ray crystallography. The basic understanding of mechanism of transcription come from these X-ray crystallographic data. Now, there are other components such as TF2B, TF2D, and TF2A which are associated in this particular core promoter region. TF2B is an important player in this process. Eventually, TF2F would be recruited, which brings in the RNA polymerase as well. And in this complex, TF2E and TF2H are associated. Now, this particular complex is known as the pre-initiation complex. Still, the RNA polymerase cannot move out, but still the minimum requirement is kind of fulfilled. When can RNA polymerase move out? When TF2H, which has a kinase activity, phosphorylate specific serine residues in the C-terminal domain of the RNA pol 2, that is the signal for RNA polymerase to move out of the start site and run along the gene body. This is the signal for RNA polymerase to begin the transcription, and thereby the initiation process is completed. So let us quickly summarize what we learned in terms of initiation. A lot of factors are associated. The key factor is TF2D, and TATA box binding protein is a subunit of TF2D. Then other factors such as TF2B and TF2A get recruited. Further, it has been shown that TF2B interacts with other core promoter elements such as BRE element. Then there is RNA pol 2 recruitment along with TF2F. It's important to note that the TBP, TF2B and DNA complex is stabilized by TF2F, which is really important for transcription initiation. Later on, TF2H is the biggest subunit of all these general transcription factor, and it has the kinase activity, so it can phosphorylate the C-terminal domain of the pol 2, which is a start signal for the RNA polymerase to leave the promoter region and run along the gene body, and thereby transcription is initiated. Now, after transcription is initiated, the polymerase gets stalled in a position just nearby the promoter, and this is known as proximally paused polymerase, and this is a problem for the transcription process. So at the first goal, polymerase cannot run the entire gene length and it requires additional help from factors. Why it cannot run the full length? There are factors such as DSF, such as negative elongation factor, which are holding the RNA polymerase movement. Now there are elongation factors such as PTEF-B or positive transcription elongation factor, which phosphorylates the NEF, the DSF and the C-terminal tail. Phosphorylated NEF or negative elongation factor dissociates from the RNA polymerase, and this is a signal for RNA polymerase to move further and get released from the stalled position. Now RNA polymerase is free to move and it moves along the gene body, transcribing the RNA. You can see the RNA is protruding out right now. So this is the overall elongation state. During the time of elongation, few important biological modifications are happening in the RNA. First of all, there would be recruitment of several enzymes collectively known as the capping enzyme, because eukaryotic mRNA has a M7G cap on the 5 prime. So there are enzymes like methyl transferases, guanyl transferase and triphosphatase which work hand in hand to bring this kind of modification in the 5 prime end of the nascent mRNA. Before even the elongation can finish and the transcription process is terminated, the M7G cap is formulated. Now why M7G cap is important? Because M7G cap protects against nuclease attack. It can help in mRNA export, and also it can interact with translation initiation factors, so it can aid in translation as well. Just like these capping enzymes, there could be other category of enzymes known as spliceosome. So this splicing enzyme ensures that the introns are spliced out and only exons are left out. So that is already happening when the elongation is in progress. So all these splicing enzymes are recruited into the C-terminal domain of the RNA polymerase and they do their job. In the termination step, there are few important events which has to happen. A polyadenylation tail has to be added. There are specific signal sequence such as AA, U, AAA. These sequence attract the protein CPCF or cleavage promoting factor. There is also CSTF, cleavage stimulating factor. Ultimately, there would be recruitment of polymerase known as polyadenylation polymerase, which is template independent, and as a result RNA polymerase would eventually be dissociated and a poly A tail would be generated. If you need to learn more about the polyadenylation process or the M7G cap formation process, you can click on the video in the I button or in the description for a greater detailed video. Anyway, let me talk to you about some landmark discoveries in context of eukaryotic transcription. Roger Kornberg, who was actually a family member of Arthur Kornberg, got the Nobel Prize in 2006 for his key contribution in the field of transcription. Two of his major contribution was to solve the 2.5 Armstrong resolution structure of the RNA polymerase, and later on in recent year, with the help of cryo-electron microscopy, they figured out the structure of entire RNA pol2 pre-initiation complex. These are two landmark papers in the field which has changed our understanding about eukaryotic transcription. All the links of these papers are provided in the description, so don't forget to take a look at those. Anyway, so far we are talking about the transcription happening in a test tube. Things are much more simpler in test tube, but things are difficult when these processes are happening inside an intact cell. Let me tell you why. Because the GTFs or general transcription factors such as the TF2 family transcription factors are the minimal requirement for transcription in a test tube. But it really doesn't enhance the transcription rate that much. In eukaryotes, in the in vivo situation, the rates are very different compared to in vitro. So what really regulates the overall rate? It turns out there are regulatory elements that can influence the transcription or rate of transcription. So this is the core promoter where the general transcription factors are recruited, and which further recruits the RNA pol too. So this is the minimal requirement for transcription to begin. Alongside these general transcription factor, there are specialized transcription factor which can be recruited into the enhancer. They can interact with other components such as mediators, which can be either, let's say, repressor or activatory complex. And based on these kind of interplay, the fate of a gene is decided, whether it would be transcribed or repressed. Let me tell you about this in a bit more details. So let's say a particular region is linearly far away from a particular gene promoter, let's say 30 kilobase pair far. But these distantly located regions can come closer if a loop is formed. So generally, enhancer promoter loop is formed where RNA polymerase can be recruited, specialized transcription factors can be recruited into the enhancer region, and it can bring out the transcription in a much stronger fashion. So there are specific methods known as 3D conformation capture, which can capture the 3D conformation of a chromatin and can tell us that which locations in the genome are coming close in 3D space and time. So these kind of method can really tell us about a contact map. In a different video, we'll talk about Hi-C in much more details. But let me tell you that, okay, if this is a 3D chromatin, the chromatin is really dynamic. At time t0, let's say a point A and B are close together, but at time t1, they might be far apart. Think about again the time t0, there might be 2 points, C and D, which are far apart. Now at time t1, they have come closer. So all these new contacts are built and old contacts are resolved. So that is why 3D chromatin is highly dynamic. It's not only within the chromatin, there could be interchromatin interactions as well. One enhancer region might be present in completely different chromosome which is influencing another particular chromatin. So that is completely possible. That's why we have to understand the transcription from a 3D or a chromatin-centric perspective. So let's begin the understanding of chromatin-centric view of eukaryotic transcription. There are many players, don't get confused, things are a bit easy. So this is a 3D cell. We zoom into the nucleus because the nucleus is the hub for transcription. Inside the nucleus, there would be genetic material densely packed like this. So in this densely packed forest of DNA, transcription factor has to find its promoter or enhancer region. So things are very complicated, jam-packed inside the nucleus. So how this problem is solved? So whenever we look at the particular nucleus, we cannot think only about the DNA, because DNA is wrapped around histone octamers forming chromatin. That is why we need a chromatin-centric view. Let's say at a promoter, general transcription factor are added and then polymerase sits on the promoter and it tries to start the transcription. But in the path of the gene body, there are several obstructions such as nucleosomes which can act like a roadblock. So how these roadblocks are overcome? That is a key essence of understanding the chromatin-centric view of transcription. It is also possible that the chromatin is wrapped around the histone so tightly that RNA polymerase or the transcription factors are not getting enough access to the promoter. So how does transcription happen in that case? How is the accessibility achieved? So let me tell you, transcription factors are not lonely beings. They always interact with other molecular players, such as the nucleosome remodeling complex. As the name suggests, they remodel the nucleosome. Sometimes they loosen up the DNA wrapping around the histones and creates accessible areas for transcription factor to bind. It also creates accessible areas for RNA polymerase. This is only one way. There are many other ways. So imagine in a 3D chromatin, there is transcription factor which cannot gain access, so it takes help from the nucleosome remodeling complex, which now also can change the chromatin accessibility. Also, that specialized transcription factor can interact with, let's say, the histone acetyl transferases, which can modify the histones in these nucleosome. Let's say they deposited some acetyl group. So overall packing of the nucleosome is now going to be changed. Acetylation of the histone would create more accessibility. The chromatin around this region would be loosely packed. That would allow the transcription machinery to be recruited into the promoter, and the RNA pol can start its job, can run along the length of the gene body. So this is one of the key essence of regulation of eukaryotic transcription. So moral of the story, transcription factor and its function is depend on what it interacts with. What are the friends of these transcription factor? Transcription factor can interact with co-activator complex, which lead to activation of transcription. It can interact with co-repressor complex, which might lead to inhibition of transcription. It can interact with histone acetyl transferase, which can lead to increased accessibility of the chromatin, which would in turn increase the probability of transcription. Then it can interact with histone deacetylase, which compacts the chromatin. It can interact with nucleosome remodeling complex, which can do plethora of things, either replace some histone, either totally kick out histone from its own location, and maybe slide nucleosome and free up new DNA regions. So a lot of possibilities are happening inside the nucleus. A combination of these steps might happen. Not all of them are necessary together. Anyway, there are specific variants of particular histones which are associated with transcription, such as H3.3 is a histone variant which is associated with actively transcribed genes. This particular histone can be found in promoters, gene bodies, and cis-regulatory elements. H3.3 ChIP-seq would be an ideal way to understand where these particular alternated histones are located in a gene body, or let's say in a regulatory element. Similarly, H2AZ is another variant of H2A histone, and it is selectively found near the transcription start site. So there are specific histones which can dictate the type of the overall rate of transcription. So question is how? How can these histone variants modulate the transcription? If you don't know about ChIP-seq, there is a video in a link in the description box. You can quickly look at what is ChIP-seq. But ChIP-seq tells you that, okay, what all particular histones or specialized proteins are located in which region of the chromatin. Anyway, so let's say, how do we know a particular gene is actively transcribed? We can also look at the serine 5-phosphate ChIP-seq. You know, all the polymerase which are active, they have to be phosphorylated in the serine 5-phosphate region. So any peak corresponding to serine 5-phosphate would show us or tell us that the transcription is active. So in this case, the pink peaks all over the gene body is showing us that the transcription is actually active. Okay, but what is the mechanism? What is so special if H3.3 or H2AZ is recruited, it would augment the transcription? It turns out that H2AZ and H3.3, when recruited, destabilize the histone-histone and DNA-histone interaction. That means chromatin conformation now changes. It opens up and helps the transcription factor to be recruited and specifically bound in a region. So it increases accessibility by destabilizing the nucleosome. So take home message, histone variants alter the interaction between histones and histone DNA. So that is why next thing that one should ask, like, who is putting these particular histone variants in place? It could be nucleosome remodeling factors or histone chaperones. It turns out that these H3.3 version of particular histones are recruited by histone chaperones, whereas H2AZ is recruited by specific remodeler complexes such as P400 or SR caps. So all of them are like SWR1 type of nucleosome remodelers. Now we have now understood why studying chromatin point of view is really important to understand transcription. Now let us be a little bit more scientific and try to ask ourselves how we can experimentally understand transcription is happening or not. Okay, transcription is fairly simple process. So if transcription happens, there would be production of RNA. So quantification of RNA would give us an essence that how much transcription is happening. One can look at processes like real-time quantitative PCR to quantify specific mRNA of interest. You can do like tens and let's say several order of 10 mRNA to look at whether some specific genes are getting expressed or not. So if you want to look at selective genes, real-time quantitative PCR is good. But what about if you want to get a holistic view or let's say a global view, then RNA sequencing is a good technique. So it tells you how gene A to gene Z, I mean, like there are so many genes that can possibly change, and RNA sequence give you an overall global picture about that. Okay, So now there are other strategies such as reporter assay, or let's say spatial information can be obtained by fluorescence in situ hybridization. In reporter assay, you start with a reporter gene. So reporter gene fluorescence would tell you whether a transcription is on or off. So if you have fluorescence, transcription is on. If it is not on, then the fluorescence would be low, or that would give you a readout that the transcription is off. So more or less, transcription would be indicated or read out by the level of fluorescence. So these are linear readouts and can be tracked in a live cell. Then spatial information about transcription can be obtained by fluorescently labeled probe. So basically, using fluorescent label probe, a particular mRNA can be detected in an in vivo situation in an intact tissue. This is known as intact tissue or cell. This is known as fluorescence in situ hybridization. So it can also tell us, okay, not only transcription is happening or not, where does the new mRNA are located. So all these informations can also be obtained by this particular technique. Now, ChIP-seq is another very useful technique for molecular biologists to understand the aspect of chromatin, because we need to understand the transcription from a chromatin-centric view. So signatures like H3K27 acetylation, H3.3, H2AZ, serine 5 phosphate combined with RNA peaks can tell us what is the level of transcription. In this case, you see H3.327 acetylation is a global acetylation or a gene activatory mark. And in this case, it is totally lying throughout the gene body. That means this particular gene or the chromatin correspond to this particular gene is very accessible. Also, H3.3 peak tells us the same story. H3.3 is found in actively transcribed genes. H2AZ combining with serine 5 phosphate ChIP-seq and RNA peak is telling us that this particular gene is actively transcribed in this given time point. So ChIP-seq is a good way of understanding various aspects of transcription. Now let us take some example of a multi-omic approach. So let us have two cell types, neurons and fibroblast. And let's see whether a gene of interest, gene A, is expressed in one cell versus other cell. So it turns out that gene A is expressed in the neuron. That means mRNA of gene A is found in neuron and not found in fibroblast. So it's kind of like a neuron specific gene. So only by looking at the RNA we get partial picture. We need to look at ataxic peaks. Ataxic is a technique which tells us how accessible the chromatin is. So in order for transcription to happen, chromatin need to be accessible. One can look at the ChIP-seq peak for a particular histone mark, or let's say a transcription factor binding. Then combine these information with RNA sequencing to get a holistic view about the transcription of a particular gene in a particular cell type or a particular context. That means multiple high throughput sequencing approaches need to be combined together to derive a conclusion. Now, if this video is too quick for you, then you can click on the I button and go to the ataxic video and try to understand how ataxic works. But anyway, we learned several ways by which we can study transcription. You can get more notes and flashcards in my Facebook page or in my YouTube community post. You can support our channel to get this kind of high quality videos all the time. You can tell your friends to support us as well. See you in next video.